Welcome to our Biomed Unleashed podcast. I'm Alisa, a student specializing in regenerative medicine at Maastricht University.
And I'm Misha, also a student at Maastricht University studying the Maastricht Science program.
Our podcast is endorsed by the Maryland Institute for Technology-Inspired Regenerative Medicine and supervised by Professor Lorenzo Moroni. We aim to make regenerative medicine accessible and understandable to everyone.
In our episodes, we invite scientists, specialists, and innovators to share their career insights and latest discoveries in this exciting field of regenerative medicine.
Today, our guest is Professor Ali Khademhosseini. He is a leading scientist in the world of bioengineering and personalized medicine.
Professor Khademhosseini is the CEO, Director, and Terasaki Distinguished Professor at the Terasaki Institute in the United States. Previously, he was a Leahy-Knight Professor at the University of California, Los Angeles, and a professor at Harvard University. Moreover, he was an associate professor at Harvard Medical School. With a PhD in bioengineering from MIT and master's and bachelor's degrees in chemical engineering from the University of Toronto, his contributions to the field of bioengineering and personalized medicine are both groundbreaking and inspiring.
In this episode, we're also going to discuss Professor Khademhosseini's successful transition from academia to industry and how it affected his future career. Ali Khademhosseini was cited more than 133,000 times and has an H index of 187. Professor Khademhosseini has been honored by more than 70 major awards at both national and international levels. Hello, Professor Khademhosseini. We're Alisa and Misha, and we greatly appreciate having you as our guest today.
Good afternoon, Professor Khademhosseini. Thank you for giving us such an incredible opportunity.
Thank you, guys. Awesome to have you visit us in sunny Los Angeles.
We would like to start from the very beginning. As we currently understand, you were born in Iran, and your family moved to Canada when you were young. And you did not speak English when you started this new life chapter. How did you experience the new environment?
Yeah, no, I think definitely there was a lot of, things changing very quickly. So, but people are resilient, so you just have to kind of go through it. But obviously going to a country not speaking the language is difficult. And, moving to a new country, making all new friends and new life is very challenging. I think the good thing is sometimes you're too young to even to really understand how difficult it is. But the good thing about being young is that you do pick up languages fast. So I think within 8-9 months, I was enough to kind of be able to communicate effectively with what I wanted. But definitely initially moving to a new country is always difficult, independent of whether you know the language or not. But good thing, my mom was around at the time, and I have a brother who was around, so made all of this transition easier.
Mm-hmm. And you often say that throughout your life you constantly seek ways to make a positive impact on society. Have you already had it since you were a child, or was it influenced by a particular person or experience?
Yeah, so I think I've always had grand kind of visions of doing great things, and I think everyone has that. Everyone growing up has their own views of what they want to do. I think that realistically being able to do that and actually accomplish something changed when I started getting exposed to research, because that's when I felt really passionate about it. And I think research is one of those things that is exciting, and if you're actually interested in doing research, and you can potentially solve important problems. So really it was a journey. I think when I was in Toronto, I did get exposed to a lot of great people, but it wasn't really until I went to MIT for my PhD and seeing people like Bob Langer that really got me thinking that, you can really follow the path that I'm following and be able to make some important contributions. So really it was a journey, but I've always been very passionate about doing things and being good at what I do, which is important. You have to be able to overcome difficulties and really persevere and overcome challenges if you want to accomplish things. And that's one of the things that I was lucky enough to do when I was growing up, and also when I went to do my graduate studies.
And talking about your passion, in previous interviews, you mentioned that during your third year at the University of Toronto, when you were getting chemical engineering degree, you had an internship in a tissue engineering lab, right? Which influenced your decision to pursue a career in tissue engineering. And could you share a specific moment from this internship that was particularly impactful for your understanding that you are passionate about it?
Yeah, I think it happened very early in the internship. So I was studying and doing lab in the lab classes, but I hadn't really done an internship in science. And the first time I got to do that and actually go inside the laboratory and read papers about bioengineering and the fact that you can take your knowledge from what you've learned in school and apply it to a whole new area of solving medical problems, to me was very fascinating. I remember back in those days before internet, you still had to go and photocopy papers from the libraries. So I spent quite a bit of time doing that, actually. I find even that concept of going to see these papers in these journal articles pretty interesting. And just being able to think that what you do in the laboratory can actually one day be able to lead to a therapy or medical device of some sort. So right from there, I got really excited about it. And I think that when you think about major things that are happened in human science and influencing human health, seeing that how the kind of things that people have made have build up to lead to where you are in your scientific career, and now you're really at this stage where you can make your own contribution. So you can read about history of science and all the discoveries and all the knowledge that has been gained over centuries, and then you're at that front and you have all that knowledge, and then you can kind of take it and then do something with it. And I found that really exciting because it just means that you're only limited by yourself and by your thinking and how much you're willing to put into something, as opposed to being limited by other factors that are outside of you. So I thought that was really exciting, and so I got super passionate right then, right from the beginning. And the tissue engineering also research was really exciting because being able to use materials and cells and biology and engineering to combine it all together to address organ shortage problem, I think is one of the most exciting things. And I still get excited about it. I have some projects in my lab that right now are trying to really make it happen, which is super exciting.
And after that, why did you decide to pursue a PhD in bioengineering in the United States exactly?
Yeah, I think I was at University of Toronto for long enough. I was there by the time I left, was there 6 years. And I think that's a long time to be in one particular place. And I always felt that if I got new experiences, went to new universities or new countries, it would benefit me. And obviously, US has a lot of great schools, and I always wanted to go to a place like MIT. I actually had my professors in Toronto were MIT alumni, so I always kind of had this impression that, wow, MIT would be such a great place to go because my professors were alumni of that university, and I had just heard about so many great things happening over there. So when I got this opportunity to apply and get admission to MIT, I think it was my top choice. And at that point, I had already done my bachelor's, and then I had done 2 years of intensive master's research program. So I felt that it was the right timing. And when I went to MIT, there's a lot of great professors over there that you can work with, including who I wound up working with, Bob Langer, who's an incredible scientist.
So you knew Professor Langer before going?
I knew of him before going. I had probably emailed him for some things, and he had responded to me. So he had, so I didn't really know him, but I felt that I know him.
Nice. And you worked more than 17 years in academia, also as a professor at Harvard University, at MIT, UCLA. And after that, you decided to build an institution that focuses on innovations based on translating the laboratory research to real-life applications. And you started doing that at the Terasaki Institute. And what motivated you to leave UCLA and start a new research organization?
I think, part of it was, again, at different times of your life, you feel like you're ready for a change. And that was the right timing. I had done basic research and just publishing papers for pretty much the sake of publishing papers for a long time. And I was starting to kind of appreciate that to really make bigger impact, you have to go beyond just publishing papers and doing work in the lab, but try to actually get something that goes to the real world. And that is something that very few people actually get to do, particularly in academia, because when you think about professors and how many of them have actually done something that has made it to the real world, there's very, very few of those people around. So I really started thinking about, how do I want to do that? One option would be to kind of stay at a university and then try to do it there. I did that for some of our early work. So when we developed some of our first technologies that we thought were translatable to the real world, we went through the university pathway of generating the intellectual property and then building the IP portfolio and then trying to license it. And that works fine, but we did find number of challenges within that model, which was typically universities are designed to really tailor for education and not built for enabling company formation and innovation aspects. So that was one challenge. And then the other thing is really lack of resources sometimes, where every time you want to do something, you have to wait for a funding opportunity to come along and be able to fund that project. And it's a very slow process. So I felt at the time that there's a lot of things that can be done better. I arguably was in some of the best places for translation, places like, of course, MIT and the Wyss Institute, which was doing a lot of really innovative things at the time. So I had some positive suggestions or experiences about what works in an academic model to induce translation, but also seen some of the challenges. So when I got this opportunity to talk to the Terasaki family, who are the benefactors of the foundation and led to the institute, I pitched the concept of trying to take the good things from that model, but also try to leave some of the things that are not designed for innovation, kind of create something that really focuses on innovation. And I think that resonated with them, and the opportunity was just the right time. It was a really good opportunity, and it was an interesting time for me to start really the next chapter of what I wanted to do.
Could you please share how this transition from academia to industry has affected you personally and as a researcher?
Yeah, I think you value different sorts of things when you are exposed to both industry and academia. For example, in academia, the currency is your publications, right? That's the only thing that really matters. Whereas in the real world, that doesn't matter at all. People care about other things. So as I went through and trying to make the transition, I also spent a year at Amazon learning about their corporate culture and how they do scaling of the different innovations that they do. And that really helped me kind of realign me to get me to think that, yes, we need to have both the ability to come up with new ideas and try to push them forward, but at the same time, most of the difficulty is actually after the idea process. Idea and even developing some preliminary data around the idea is kind of easy, but really taking it all the way to something that becomes a commercial viable product that's going to be used by people is a very, very difficult and different thing. So it just it makes you having one foot in each of those worlds makes you understand the challenges and advantages that each of those places has, and allows you to some degree to appreciate both of those ecosystems. And I think the most difficult thing is to try to navigate between the two, to be able to communicate with both types of communities, and at the same time pick what's best, from academia that's ready for translation, and then use the industry mindset to get it from a laboratory to a commercial viable product. So for me, it's been really interesting, and it allowed me to not only realign but learn so much different things as well, which I continue to do. And I think that's really exciting, because if you're just doing same thing over and over, then it just for many years at a time, no matter how exciting it is, then at some point it becomes routine. And I felt that sometimes the research part just started becoming routine, but now really trying to focus on the challenge of taking that to the real world, it makes it a lot more exciting again.
But for many people, it's very hard to make big changes in their lives. Was it like that for you as well?
I think it's always hard to make big changes. There's always risk and not knowing how it's going to affect you, and being successful. I think really at the end of the day, it comes to what you think is success. What is it that you value at different stages of your life? In particular stages of my career, I've thought about, let's say, academic metrics. What is my academic metrics, and how do I try to push them forward and be very strong in those? And at other points, then that metrics didn't really matter anymore. I'm like, okay, I've done all I want with that, but now what? So, and I think, once you get to the certain stage of your life where you also have some stability in your career and in your, financial situations, then also making drastic changes, it's a little bit de-risked as well, because that you can always go back and, say that I've been a great professor, I can do it again and, be okay, right? So, but to me, the challenge part of learning new things and trying to increase your level of impact outweighs the risk part that is associated with any time you make a big change.
And you are also Chief Executive Officer and Director at the Terasaki Institute here, the main mission of which is to create personalized practical solutions to enhance the health of individuals. You're also a leader in applying bioengineering solutions to precision medicine. And before we go further, could you please tell us what precision medicine is?
Yeah, so it really, in my mind, is a little bit different than perhaps in some other standard definitions. To me, it's going beyond the standard genomics and proteomics and those kinds of omic world to things that are much broader in terms of how do we personalize medical treatments to people. So, for example, like when I think about this institute, the institute was built from the contributions that Dr. Paul Terasaki made for matching recipients and donors for organ transplantation, right? So that really is a personalized therapy. The transplanted tissue has to match the recipients. So that's just one example. To me, tissue engineering itself is arguably personalized because your organ that you're building has to match the site and the genetics and all that sorts of things. To me, when you're making a medical device, that medical device should be tailored for that individual. It should perhaps be the right size, the right way of responding to that person's environment. So to me, it's beyond just the omics world. It's actually a whole bigger picture of how do we get the right therapy to the right person, whether that's through molecular approaches of omics, or whether that's through personalized cells or personalized materials, or being able to model a person's physiology on a chip, like some of these organ on a chip models that we have, or be able to make a tissue that is tailored to the person. So to me, it's a broader way of really providing that personalization for any therapeutics that you're doing.
And what influenced you to choose it as a main vector in your work?
I think part of it is, of course, what you know should derive what you're working on to some degree. So my background has been in materials and cells and regenerative medicine and things like that. So obviously I look at the problems with that mindset. But the other thing is that, of course, the impact that it has and the challenges that exist right now in existing medical practices and trying to address them. So it's a combination of the need as well as the push and the pull, the need and the expertise that we have.
Mm-hmm. And what do you consider your first success in personalized medicine research?
Yeah, so obviously when it comes to translational work, I think there are, the success that has made it to the clinic and is being used is a product that we have, which is to some degree is also personalized, because even though it's a medical device, is a material that conforms to the shape of let's say, blood vessel or an aneurysm. So it's a material that is what they call an embolic. It's a material that's used to plug blood vessels or plug blood vessels for internal bleeding or aneurysms before they open up. So I would say that's kind of the one that has made the most clinical impact so far, because it's actually used in the hospitals right now. And it's an early example of what I would say personalized, because it's a smart enough material that it's shear thinning, so it can, like a toothpaste, fill the void that it exists. But at the same time, it doesn't necessarily have a lot of biological cues to be able to tailor regeneration or other sorts of things. So it's a version 1 personalized therapeutic, but there's obviously a lot more advanced things that one can make as well.
And you talked about organs on a chip. We think that the systems are a fascinating area of research, and could you please explain how the systems work?
Well, organs on a chip are basically models of human tissue outside of humans, by taking someone's cells, being able to bring those cells together to make miniaturized version of a person's tissue types, in a way that those little tissues represent and function the same way as human tissues would in the body. So the advantages that it has is that one is that those cells are personalized, so you actually can tailor that tissue to an individual. The other thing is that the cells are more functional, so instead of having cells in a dish that is not representative of the complexity of tissue, you can have different cell types, you can have different sort of organization or architecture that is associated with functional tissue. You can even have different tissue types. So you get more, not only organs, but multiple different organs represented. And the way it pushes existing technologies forward is that you can get better functionality of these cells, so they mimic the behavior of what you see in the body better, and that hopefully leads to better outcomes in terms of predicting whether something is toxic or whether some compound can actually be able to treat a disease, let's say. So those are some general reasons why organs on a chip has become exciting to people.
And you said that on one chip you can mimic different tissues. And in previous interviews, you also talked about human on a chip system, which mimics, as far as I understand, all vital human organs. As far as I understand, it's not created yet. And when do you believe it will be possible, if possible, to successfully develop this human on a chip system?
Yeah, I think Realistically, most of these systems only need to be as complex as the question that you're asking it to answer. So if your question can be answered with one cell or one tissue type, or just a single organ type, then that's the kind of complexity that you need. I think there's very few kind of questions that require all the tissue types to be present in the same system as opposed to individually. So to some degree, I think we may not need the whole complexity of the entire human tissues all interconnected on the single chip to ask a lot of important questions. So that's, so you may not need it. You may not need the whole complexity of having it all in the same chip. So that's kind of what I'm kind of hoping, because obviously every time you add complexity, it just makes things a lot more difficult. And as I've become more realistic about what it takes to take things to the real world, I really start to admire simplicity. The simpler the approaches, the more reproducible they will be, the more cost-effective they will be, and that at the end makes the translation easier.
And talking about difficulties, I know that it's possible to control the systems like temperature, pH, etc. And what are some of the challenges that you're encountering in controlling them?
Yeah, I think some of those simple processes like temperature, oxygen, those are rather simpler to control. I think when you're thinking about biological control, for example, having the cell be exposed to exactly the right environment that it needs to do a particular behavior, that will be a lot more difficult because the cells require such complex surrounding environment, and being able to make all of that happen the right way is extremely difficult. So I think those are the challenges, the biological complexity of using the extracellular environment to drive cell fate decisions. And that's why a lot of times people are starting to really think about ways of understanding how cell really functions at the genetic level, and what are the key triggering mechanisms are, the key transcription factors, or the key places where you can come in with a particular, let's say, gene and be able to really drive that outcome that you're looking for. So I think that's kind of why technologies like, this mRNA, modified RNA, or things like that have become more and more interesting, because you don't need to control the surrounding of the cell to the same degree that is controlled during, let's say, embryonic development.
Mm-hmm. And as far as we understand, there is a need for organ-on-chip systems because static two-dimensional cell cultures and animal models that are currently used cannot recreate the complexity of human physiology. And do you think such systems will be able to fully replace the animal models one day?
Yeah, definitely. Replacing animal models one day is not going to be a problem. The only question is when that one day is, right? And obviously, every time you're talking about a science project replacing standard practice in the real world, that's where really the big challenges are. But theoretically, there's no reason why we can't make these human-based tissues in the models that we're talking about, and there's a lot of logic about them being more predictive than the existing models. So I think that the science is there, the validation is more and more proven on a daily basis, but it's just a matter of taking that and adding so much more validation, regulatory, and really firepower behind all of this stuff so that it can slowly replace the existing animal models that we have. Because every time you've got a process that's developed and something that people are comfortable with, it's very, very difficult to have them do something different.
Mm-hmm. And if we talk about hydrogels, which are polymers that can absorb large amounts of water, and they play an important role in regenerative medicine and tissue engineering. And from what we understand, there are micro and nano hydrogels, and they're very small, but they have a large surface area. And they can deliver drugs or cells to body parts that are not accessible to larger hydrogels. And we know that you have done a groundbreaking work with this micro-nano composite hydrogels. And could you please explain what is the difference between this micro and nano composite hydrogels and how to use them?
Yeah, so, and definitely micro-nano is a very broad kind of area. I think in terms of hydrogel and using them for delivery applications of cells or drugs in different parts of the body, there's a bunch of different ways to do it and a bunch of different ways that those materials are kind of can be used. So some simple approaches are being able to load your drug inside a material that is a hydrogel but is made from nanoscale materials that come together to form that hydrogel materials. And those nanoscale materials can also interact with the drug and then be able to keep the drug local and slowly release it into the surrounding environment. And depending on how you deliver that hydrogel, whether that's something that you would inject, that's something that you would deliver through a catheter in a particular place in the body, that's something that you can put in the needle part of little microneedle arrays that you can put on your skin, or that's something that you can make into little microparticles that you can kind of put inside the blood vessel and have go around. There's different ways to get those things to work. But the thing that gets us excited about all of this is that the hydrogel materials themselves have a lot of features that you want for materials that are going to be in the body, so we can make them to interact well with blood and other components. We can make them to have tunable behavior, so then they go away over short period or long periods based on what the drug requires, and a variety of other tunable characteristics. So that's why we think that whole concept has lots of applications in many different therapeutic things that we want to do.
And you mentioned that you're working with biomedical devices, and we read that you made contact lenses to treat dry eye syndrome. Can you please provide some other examples of these devices, maybe which will come next?
Yeah, so we're very much interested in wearables of different sorts. So obviously you mentioned contact lenses, so contact lenses that would not only sense what is wrong with the eye, but be able to deliver drug. So perhaps things that can sense things beyond just eye-related things, whether we can sense glucose or mental health molecules or other sorts of things. So that's kind of one category of these wearables. There's a lot of interest on patches that you can have on your body, and these patches can, again, sense what is let's say what's in your sweat, or can have a little needle that can sense what's in the interstitial fluid right underneath your skin, and then from there be able to say, okay, you may have particular glucose levels, you may have particular serotonin levels, particular cortisol levels, and then based on that, not only give you information, not only store that information, not only be perhaps integrated with AI algorithms to really understand what's happening inside your body, but also be able to deliver potentially therapeutics based on what you're seeing, and it's communicating in real time with the cloud and with the doctor's office and a variety of things like that. So we think that there's a lot of opportunity to take those devices and apply it to virtually every kind of disease that you can think about, because in most cases, ability to monitor in real time, deliver interventions in real time, and be able to do it in a way that is secure and connected with the doctor, and potentially with first aid, 911 type things. There's a lot of interesting kind of applications like that in wearables that come in all sorts of shapes and sizes.
Okay. Mm-hmm. And I think for today, our last question. What would you do if not tissue engineering?
Yeah, scientifically or just career-wise? Or
I think career-wise.
Yeah. I think, definitely if I wasn't going to do, like, let's say, tissue engineering, materials, science-y type stuff, and I wanted to do something again, I probably would have thought more about other areas at the interface between biology and engineering, maybe like things like synthetic biology or things like that, maybe more computational things, combining things with AI, I don't know, things like that. I think career-wise, I think that's a really interesting question. I definitely like to work, with teams and with lots of other people. So I probably would have tried to be in some sort of managerial kind of role if I didn't do scientific things. I think I generally like those things more, to kind of be able to organize initiatives and push them forward and things. So those are some areas, yeah.
But I think right now you're also combining managing work with research, right?
Yeah. So it would be more like if I didn't do research managerial, I would just do managerial.
Okay.
Yeah.
Okay.
Professor Khademhosseini, thank you so much.
Really appreciate your time. Yeah. I hope you guys have a wonderful visit to US.
Yes. Thank you very much.
Thank you very much.
It was very exciting to listen to your story. Thank you. Thank you so much for listening. We hope you enjoyed this episode of Biomed Unleashed podcast and will stay with us for the next ones.